Welcome everybody again to another episode of the ideaXme show, that profiles the humans behind the really big ideas that are shaping our world, inspiring the future and future creators, and for all those really great stories. I'm Ira Pastor. I'm your health and longevity ambassador along for this journey. So today, as we continue our discussion pertaining to the so-called biological hierarchy of life, we are going to journey into what I refer to as the metabolic architecture of the cell, and all the fascinating dynamics that surround our genome and allow for the information that's encoded within to ultimately impact the ongoing physiological states of our health, our transition into disease, and ultimately aging. And in doing so, we will segue into a discussion around a category of biologic entities, broadly defined as ribonucleic acids, or RNA, which are polymeric molecules essential in various biologic roles, including coding, decoding, regulation, and expression of genes. Most cellular organisms use one of the primary forms of RNA, namely messenger RNA, to convey genetic information from our DNA to the synthesis of specific proteins. And that process is extremely integral in the appropriate gene expression across our 200 plus different cell lineages. That's why a cardiomyocyte has different sets of proteins expressed than a neuron, than epithelial cells, and so forth. RNA silencing or RNA interference ultimately talks about the process which protein production from a gene is turned off, and the term RNA therapeutics means either the use of one of the other forms of RNA that exist. We get into these regulatory RNAs, small interfering RNAs, microRNAs, and so forth, directly as therapeutic agents, or the modulation of these processes with more traditional small molecule drugs, for example. And because RNA is so central to all biologic processes, there are numerous potential avenues for addressing human disorders at the RNA level, including most chronic degenerative diseases, infections, and even the process of aging themselves. So that's why I'm so excited that today we have our guest joining us, who is an expert in this very unique domain of biology, and who's going to take us further into the topic. And that's Professor Lorna Harries, a professor of molecular genetics at the University of Exeter Medical School. Dr. Harries is a PhD in molecular genetics from University College London, and set up the RNA-mediated mechanisms of disease group at the University of Exeter Medical School in 2006. Her team has a special focus on how RNA biology influences both normal and disease states, using large-scale omics approaches down to detailed assessment of specific mechanisms of specific genes. Dr. Harries' group leads the group's investigation of how and why we age, and the reason age is such a major risk factor in a variety of chronic degenerative diseases like type 2 diabetes. Her goal is to use the information the group uncovers to develop a broad range, a new generation of anti-degenerative drugs. And as mentioned, as many age-related diseases have common roots, causes, and mechanisms which arise from the failure of a few sort of central control mechanisms that decline in efficiency as we age, the Harries lab feels that focusing and targeting them will ultimately be able to allow us to target multiple age-related diseases at once. Dr. Harries has written over 90 peer-reviewed articles. She was awarded the Diabetes UK R&D Lawrence Prize Lectureship in 2011. She's the coordinator of the annual University of Exeter Medical School Men in White School Outreach with Dr. John Chilton, which is an event that brings in Year 9 students beyond from school stretching from South Devon to North Somerset, and gives them hands-on experience in the work of a research laboratory. And Dr. Harries is a STEMnet ambassador and also Exeter Catalyst champion for public engagement. All that being said, Dr. Harries, thank you for taking the time to come on the show today.
You're welcome.
Typically, we start out by giving our guests the floor just to talk a little bit, about themselves. Obviously, a lot of people are familiar with you in the longevity biotech community, but for those unfamiliar with you, could you just go into a little bit of your backgrounds, or where you grew up, how you got interested in science, in medicine, and ultimately how you developed an interest in this really sort of integral set of processes behind health, disease, and aging?
Yeah, sure. So I was born in the Midlands in, a very long time ago. I'm not going to tell you exactly how long, but suffice it to say that I'm now very interested in aging. So actually, as I was growing up, I've always been fascinated by complexity and by sort of how things work. I was always the kid in the playground that wanted to know how that is working and why things are a certain way. So I'm actually a failed vet, would you believe? So my plan was to go into veterinary science. James Herrick has a lot to answer for, I think. There's a whole lot of us from my generation who were very keen on that idea. And then it became quite evident to me as I kind of looked a little bit more into it that a lifetime of clipping old ladies' budgies' toenails may not be the most exciting way to spend your day. And I got interested in genetics. So this was back in, 1988, so a long time ago. And genetics at that time was a really nascent field. There was not much known about it. It was viewed as something that was really complicated, hard to understand. And there were about 30 people enrolled on my undergraduate degree in genetics back then. Now I think the average intake is about 210 per year, so we've grown somewhat. It's now, and the potential implications and the impact of this field has really become very evident. So basically, I got bitten by the bug. But my plan was to do genetics as a stepping stone actually into veterinary college. But as I say, I just got so interested in it. It's a fascinating subject. So the more you delve into it, the more there is to know, and you've never finished. So I did my degree. That was at University College London. I then stayed there for another 3 years and did a PhD in molecular genetics at UCL again. That was in mouse genetics actually. So I was interested. This is in the days before we had the human genome. So we were interested in mapping genes. Using sort of linkage really to be able to do that. So I did my degree and my PhD. After my PhD, when I graduated, I moved up to Scotland, and I started a postdoc in the kind of pharmacogenetics field, so looking at polymorphisms in drug metabolizing enzymes and looking how that impacted on cancer actually. And then I had my family and decided that actually I didn't want to do science, I wanted to just be a mum. And I took 6 years out. But the plan was I was going to stay home till my youngest was at secondary school, and I made it till he was 2, and I was fine with it. So yeah, by that time we'd relocated down to the southeast, and then shortly after that down to the southwest where I am now. And I basically turned up at the medical school and targeted anybody who looked like they might be able to give me something interesting to do other than talk about nappies and washing powder. And was lucky enough to land a postdoc position in the laboratories of Professor Andrew Hattersley and Professor Sian Ellard here. They're interested in monogenic diabetes. So that's really where my interest in RNA started. So I was in a whole sea of people who work on DNA. So in my institution, there's a lot of people who are gene hunters. They're looking at genetic association studies for all sorts of things, from diabetes to heart disease to hypertension to height, all sorts of things. And what struck me very quickly is that although people are quite good at identifying loci associated with various disorders, they are less good at translating those findings into something that we can use. So how it actually works, what does that SNP do? And it became very evident quickly that actually a good deal of those SNPs, a good deal of the genetic variation that's associated with common chronic disease is in the non-coding regions of the genome. It's not usually in bits that make protein. So that's where my interest in RNA came. And in the sort of 3 or 4 years that I was postdocing in Andrew and Sian's lab, they basically let me go off on my own track and look at the effects of some of the mutations that cause this early onset diabetes, this inherited diabetes, at the level of RNA. And that's kind of where I learned my trade. So I started off in 2006, as you say, I started my group, and we had then had a really broad scale interest in how alternative splicing, so the production of multiple isoforms from one coding unit, could impact on the phenotype of mutations. The interest in aging, so I still have an interest in diabetes. We're interested in how splicing impacts on cell identity at the moment, and we've got some cool stuff coming out on that. But the majority of the group at the moment are working, as you said, on aging and on the impact of RNA biology on that. So that's where we are.
Excellent, excellent. I really appreciate that overview, and it leads me into, you set it up perfectly for me. Before we get into some of these individual topics that you're focused on, I want to just highlight one thing. A couple of weeks ago, you had this wonderful write-up in Nature magazine that was entitled, "It's time for scientists to shout about RNA therapies."
I think so.
Yeah. the sort of tagline, it's a source of treatment, gene editing's getting all the attention, time to change. And, I think I grew up in the traditional pharma industry. I've seen, things take time, of course. I'm here in Philadelphia where, one of the first gene therapies a couple decades ago was once tried, and, they were failed, but it came back. And so things like gene therapy, stem cells, they come in waves. And, RNA therapies are one of these things that, it's been tried over time, but I think, as you say in the article, I think we're probably now at the curve where things are happening. Talk a little bit, if you would, just about your vision of sort of the history, and then why you think we need to shout.
Well, I think there's been a lot of interest in the gene editing. So when people talk about sort of gene therapy, conventionally people have been talking about editing at the level of DNA. And that's quite hard to do in people for lots of reasons, although there are things moving apace. This new form of CRISPR which came out a couple of weeks ago, which people are quite excited about. But my view is that RNA therapies are actually a little bit more analogous to a conventional therapy. It's something that you take to get rid of or to deal with whatever disease you have. So you've got your genes, they accrue mutations, either sort of you have them from birth as in the form of genetic disease, or they're things which accrue in tissues as you get older for things like cancer, dementia, that sort of thing. But I'm just thinking, if we can deal with the output of the genes, if we can deal with the consequences of those mutations, then we might not have to worry so much about dealing with the actual sequence at the level of DNA. When obviously, if you're looking at a disease where this is something that's happened during someone's life course, how you're going to address that by targeting that variant in every cell or every necessary cell in that person's body is quite a big ask. Whereas dealing with the consequences of that in terms of the output of those genes might actually be lower hanging fruit. So that's my view. And I think over the past few years, the technologies to be able to both construct novel RNA therapies that are less toxic and new ways of delivering them, we've just started to nudge into that arena now, and I think it has amazing potential.
Excellent. Moving on from that, so your group, as you mentioned, one of the core focuses on this RNA theme is the topic of splicing or alternate splicing, where, and I apologize, normally I don't ask our guests to give a lecture, but you have this process. We have precursor messenger RNA. It's transformed into a mature form. There's introns or non-coding regions, exons that have to be cut out, and there are these unique machineries in the cells, these spliceosomes, and then also you write a lot about the splicing factors, which are a key part of sort of the theme in what happens here. Could you briefly, I don't want to, you never actually a lecture, I don't want to have to take your time for that, but if you could just give us the basics on the spliceosome and the splicing factors and how you see that in sort of the tentacles for that reaching out into all different forms of disruptive, in other words, when systems disruptive, why that leads to certain diseases.
Sure. Okay. So my textbook definition, we have about, I think at the latest count, about 15,000 genes, and we make about 250,000 different proteins. So it purely stands to reason that each gene must be doing more than one thing. So how I explain this to my undergraduates is that you can think of your gene a little bit like a recipe book, right? So that recipe book has recipes. Let's say it makes cake. And that recipe book will have different recipes for different cakes in it. So let's say you have chapter 1, which is a little bit analogous to say exon 1, which says make your cake mix. Chapter 2 might say add chocolate. Chapter 3 might say bake your cake. So you can see that if you put that chocolate in, you're going to get a chocolate cake. If you don't put the chocolate in, you're going to get a vanilla sponge. So it's a way that the sort of mix and match of the exons is a way that the cell can fine-tune the output of that gene to the requirements of the cell. So it's one of the fundamentals of cellular stress response, actually. So it's one of the ways by which your cell can basically change its output, change what it does in accordance to what it needs from the environment, be that the internal environment or the external environment indeed. So We make, each gene makes about 3 isoforms or thereabouts, although they're extreme. So there's a brain gene in humans that makes about 2,000. And my favourite one is a fruit fly gene that makes 38,000 different forms from one gene, which I think is just mind-blowing. So the ability to do that is one of the fundamental things that actually keeps your cells healthy. It's also one of the fundamental things that actually brings about tissue identity and cell identity. So that makes a heart cell and the kidney cell a kidney cell. So there are 2 types of splicing. There's what we would call constitutive splicing, which is the cutting and pasting. This is the stuff that happens to every gene that has more than 1 exon anyway. So it's the process by which we remove bits we don't need. And it's regulated, but it happens across the board in all conditions and is necessary, but not all that interesting for someone like me who's interested in regulation. The other class of splicing is something called alternative splicing. And that is basically the ability to change the output, to change which form of the RNA you make according to what you need. And that is regulated by, as you say, it's regulated on 2 levels. It's regulated by the sequences around those exons. So you've got your introns and your exons in your genes, and there are DNA sequences that translate into RNA sequences in your pre-RNA. And those bind the regulatory machinery. So there are basically 2 flavors of splicing regulators. You have things which activate splice sites and things that inhibit splice sites. And those collectively are called splicing factors. Okay. So the activators are serine, arginine-rich proteins or SR proteins and others, which usually, but not always, there's never a complete rule for RNA biology. It does its own thing. But usually SR proteins activate. And the other class are heterogeneous nuclear ribonucleoprotein particles of HNRMPs. I always get that wrong. And they are usually, but not always, splicing inhibitors. So if you can imagine, you've got a splice site, which determines whether or not that particular bit of exon is included or excluded. That decision is made by a competitive binding of activators and inhibitors. And that's unique to every splice site, a different bunch for every splice site. So you can see it's an enormously complex thing.
Absolutely.
And you can also see that levels of those splicing factors is going to be really important for whether or not that gene is spliced the way it's meant to. So you can imagine that when things that change the balance of those splicing factors or things that change the sequences that they bind to are going to be really fundamental in keeping the body healthy.
Excellent.
I hope that explains it.
No, perfectly. I love the analogy. I like pies, but
cake is good.
I enjoy the cake analogy. No, that's wonderful.
For someone who works on diabetes, I don't know whether cake is entirely appropriate, but I think there's always a time for cake.
That was wonderful, and so that's going to take me to the next question. So on the show, obviously we've talked about aging in the past, and we've talked about senescence, which on previous shows, talk about this process by which either internal or external stresses to cells can cause a cell to irreversibly stop dividing and enter this unique stage of growth arrest. That has, it's a sort of a double-edged sword in the sense that it's a very important mechanism for tumor prevention, for wound healing, for embryonic formation, but at the same time, there's some downstream components for chronic senescence and secreted factors. A lot of the longevity biotech community has been heavily focused in sort of the world of senolytics, of ways we can permanently destroy a la modified chemotherapy type products and so forth, these type of cells. But it seems in your research, you've taken a decidedly, and in my opinion, a slightly more interesting direction, sort of this field of xenomorphics, where we're not looking at destroying more stuff, but looking at how we can change the gene expression using some of these tools, the splicing factors, compounds that stimulate different splicing factors in changing a senescent cell, modifying it and turning it back into a young, healthy cell. Can you talk a little bit about this? Because this is utterly fascinating work. Yeah,
sure. So we like to swim against the tide. So as I say, I'm coming from a splicing background. Where this came from was a big study we did, back in about 2009, 2010, where we basically took about 800 people, a blood sample from each of those, and we produced a transcriptome for all of them, a whole transcriptome, and we asked the question, what changes as you get older? So what first thing we found actually was really surprising to us was that actually not much changes at the level of total gene expression. You don't about 2 to 3% of your genome, which was a bit of a surprise. And then we looked at what those genes do. And what we found was that we looked at, basically we classified them by what they did. What we didn't see when we did that were telomeres. We didn't see oxidative stress. We didn't see mitochondria. We didn't see DNA damage. We didn't see any of the things that we were expecting to. Okay. What we did see was that there were 7 pathways that were majorly dysregulated in our older folks. Okay. Just 7. And 6 of those were related directly to splicing. Huh. The 7th was chromatin, which I would argue is also gene expression. But yeah, so that's where the splicing came in. And obviously, when we got those results, I was absolutely delighted because that's what we do. Mm-hmm. So had it been anything else, we probably wouldn't have done anything else with it. So that was with a population of blood samples from humans, from people. Now, working with people is obviously the gold standard because things you find are immediately translatable and transferable. But working with people when you're working with gene expression is a real challenge because gene expression is really labile. Everything changes it from, the time of day, the circadian rhythms, what people have been eating, drinking, the exercise that they've been doing, their lifestyles, everything changes it. So as we age, your blood cell proportions change as well. So we couldn't be 100% sure that what we were seeing were changes in blood cell subtypes. Okay. So one of the things we needed to do was to take it in vitro. So we generated a whole series of different senescent cell models. Basically, it's a replicative senescence, not a stress-induced senescence. So we've basically taken a young cell, mm-hmm. These are all human primaries, and we basically made them senescent in vitro by culturing them forever, it seems. Sure. Some of the cell types senesce very quickly, some of them, oh dear, it's been months and months. So we spend a lot of time in tissue culture, but we've now got several different types of senescent cells where we've looked at, obviously, fibroblasts, because everyone looks at fibroblasts, but both lung and skin. We've looked at cardiomyocytes, endothelial cells, astrocytes, articular chondrocytes, what else? Bronchial epithelia. We've also looked at some fibroblasts from folks with things like Hutchinson-Gilford progeria and Werner syndrome. Mm-hmm. So we've got senescent cell models of an awful lot of interesting cells. First thing we did was to basically ask the same question. So we have a portfolio of splicing factors in particular, and they were what changed. When we look at the things which change most, that's what happens. Okay. And we looked at what happened to those in our senescent cells, and lo and behold, they are majorly deregulated in practically everything, everything we've looked at. These things are tissue specific, so you don't always see exactly the same ones that have changed, but it's always a feature. So it might be a different collection of them, which is different, but they're always different. We also looked at splicing patterns. So the consequence of changing splicing factor levels is dysregulated splicing. So in our populations, we saw this in the human blood. We also have seen it in everything we've looked at in terms of the different tissue types. And that's where it got really interesting. So, the $1 million question for someone like me when you see those changes is, okay, you've got down regulation usually of splicing factors in senescent cells. So what happens when you switch them back on? Mm-hmm. So where that came about was I'd found a couple of reports in the literature. There's very, very little known about the regulation of splicing factors. For something which is so fundamental, not many people are actually working on how they're regulated. So we started off with small molecules. And we've used a couple of different types of small molecules. So we've used things based on resveratrol. We've also used things which are based on hydrogen sulfide. And the reason for both of those was because both of those things have been reported a lot in the literature to be associated with improvement in health span measures. Okay. And there were also a few reports in the literature that they could regulate splicing factors, but it really hadn't been looked at. That was the first thing we did. And we found that actually both of those small molecule classes were quite good at regulating splicing factors. They turn them back up. And when you do that, that was the point of the experiment, was just to demonstrate that you could actually just switch them back on. Mm-hmm. To my utter amazement, when we did that, what happened is the cells effectively rejuvenated. So they, what you saw is the levels of senescent cells in the cultures dropped. They started to proliferate again. In the case of some of the small molecules, they proliferate, and some of them they don't. and I'll come on to that in a minute. We saw that the SAS was attenuated. You see alterations in the output of the cells. In the case of the resveratrol and the analogues thereof, the telomeres were now long. So basically, for every parameter, or most of the parameters that you would classically associate with senescence, we've turned it back. Some of them are a little bit more selective, so some of the small molecules are a little bit more selective. So you see some aspects reversed, but not others. So you see the senescence reversed, you see the SAS attenuated, but you don't see proliferation. Now that's actually quite useful in a therapeutic setting because these are still aged cells. The cells still have a mutation load, so you don't necessarily want them to grow. Okay, sure. So that's kind of where we started off. Like any scientist, I'm interested in why that happened, why those splicing factors were switched off in the first place. So we kind of thought a little bit about how that might be, and knowing what we know about splicing factors and that they are part of the molecular stress response, we figured that sometimes the signaling pathways that confer the cell stress from the outside of the cell to the inside might be important in this. What came about then, there was a paper from a friend of mine, Kathy Slack, who's now at Aston, who had implicated AKT signaling and ERK signaling in lifespan in fruit flies. So what we did, we thought, well, let's have a look when we mess with those pathways in our cells. And what happens when you mess with those pathways in the cells is that you can mimic the effects of the small molecules. You can effectively rejuvenate the cells by hitting those pathways in isolation, one or the other, with inhibitors. If you hit them both together, all sorts of strange stuff happens. But if you do it one at a time, you can basically you can replicate the findings. But those pathways are difficult to work with because they regulate a million things, and they talk to all the other pathways. And there were some dose response effects and some auto-regulation. So if you put low level in, you rejuvenate the cells, you regulate splicing factors nicely, and cells proliferate. If you put too much in, the splicing factors go the other way, and the cells stop dividing. It's because there's a feedback loop. So we've been characterizing that. So we wanted to know what the very downstream effectors were. So we went looking, and we have isolated two genes at the bottom, one at the bottom of AKT and one at the bottom of ERK, which look like they're doing that. So the one at the bottom of AKT is FOXO1, which is a really well-known longevity gene, but no one had ever really tied it in with splicing before. It's never been reported as a splicing regulator, master regulator of splicing factors, which it is. The other one was a gene called ETV6, which is very underreported in the literature. It's known to be a translocation partner in one of the leukemias, but nothing else really known about it. But ETV6 is also a master regulator of splicing factors, and these are negative regulators. So this is our kind of novel finding, that they're negative regulators of splicing factors. When you knock those genes down in senescent fibroblasts, we haven't done the other cell types yet, but we're going to, but in fibroblasts at least, when you knock them down, you can rejuvenate the cells quite nicely. We've shown this with siRNA, but we've also shown it with morpholino oligonucleotides to get it past our reviewers and to ensure that we're looking specifically at what we think we are. They wanted us to do it by TNF, and we did, and it works. So we're now looking at how and why that's doing that. And where the therapeutics come in is that these things are working by moderating your RNA isoform portfolio. So if you can identify the upstream master regulators or the downstream consequences of that, or both, you can intervene. So that's where we're headed now.
Excellent, completely outstanding. And you know, listening, as you said at the beginning, you were in your intro how you were so interested in complexity. It's clearly complex, and I just, you know, I take my hat off to you for many things, but to I can see why 99% of people don't get involved in this stuff. And
that's lucky for me.
I'm fascinated by listening. I could listen to you talk for a long time about this, but I'm glad you're focusing on it, because there's clearly, you know, evolution put all this stuff together in this way, and to really understand it is the way you do is beyond fascinating.
Well, I wouldn't say I understand all of it. You know, the thing with RNA biology, actually, I suspect, like most fundamental human genetics, is the more you think you understand it, the less you actually probably know.
Oh well, but nonetheless, it's completely fascinating work. You know, leaping from that now into diabetes type 2.
Yeah.
You know, we've done a couple shows in the past on type 1 diabetes,
Yeah.
which obviously major problem, but you're focusing on the bigger of the two, hundreds of millions of people.
Yeah, it's a big problem.
It's a big problem, not just endocrine, cardiovascular, connected
Everything.
Alzheimer's,
and dementia these days, you know,
Absolutely.
there are links with cognitive function. So.
Absolutely. You know, you've been studying, once again, messenger RNA, you've been looking at the genes implicated in susceptibility, and at the same time, you've done a lot of very interesting study in terms of how various factors affect differentiation status of different cells and pancreas and so forth. Once again, a fascinating domain of work. Can you take us into that a little bit, because it is one of our major killers that we don't have a lot of answers for.
So this came about, so obviously I'm in, so the splicing and the splicing factors and the RNA regulatory world is going to impact on everything, right? So I got interested, so obviously I started off in diabetes. I kind of, you know, took a side swipe into the world of aging, and we're doing both in parallel. But actually, when people ask me what my group do, I don't say we're an aging biology group or we're a diabetes group. We are a mechanism group.
Mm-hmm.
So, and those mechanisms, as you alluded to earlier, are shared between all sorts of things. So the cell identity stuff, that came about because of a lecture I sat in about 10 years ago, where someone reported that, you know, people have always said with both type 1 and type 2, but more for type 2, that when you have type 2, that your beta cells die. And then someone stood up and said, well, actually, some of them don't. Some of them turn into other types of cells, and in mice, that's alpha cells. And that was interesting to me because, again, it's a complex question and one that not many people were looking at. And I just thought it was worth looking at and was interesting. So, and given what we know that a type 2 diabetic microenvironment, the soup that those cells are sitting in, is really stressful for cells. You've got high glucose. Not only do you have high glucose, you quite often have swings in glucose. So you might have high and low glucose. You have the inflammatory milieu that's involved in type 2. You have the hypoxia. Quite a lot of folks who've got type 2 diabetes have sleep apnea. So they will have times when their oxygen levels are really quite low. And also the dysregulated lipid that your cells are exposed to. So again, this was one of those kind of serendipitous things where the actual, you know, I won't lie, the original point of the experiment that we set up was to see what happened when you put these things on cells, on pancreatic beta cells, on human pancreatic beta cells, what the effect of swings in glucose and lipid and hypoxia and cytokines was on the cells. So we were just planning to look for changes in gene expression, really, that, you know, in terms of total gene expression, in terms of splicing, in terms of microRNA. That was the plan. When we did the work, what we noticed is that some of our beta cells were no longer beta cells. So this isn't a cell culture. This isn't a cell culture that is supposed to just be beta cells.
Sure.
When we look at them, we have some delta cells, which is unusual. So we went looking for delta cells in people with type 2 and also type 1. And what we see is that certainly with people with type 1 who have a lot of hypo and hyperglycemia events and a lot of inflammation, not so much the disrupted lipid and not so much the disrupted oxygen, but what you do see is there's a lot more delta cells, so somatostatin positive cells than you would expect. So about 25%. So normally it's about 3-4% in a normal islet. In folks with type 1, it's about 25%. In type 2, again, you see a lot more delta cells than you would expect by chance. So we went hunting for what's going on there. And what we found is that actually, when you look at the transcriptomic output of those cultures where you've got more green cells, there's an awful lot of dysregulated splicing. And this is one of those sort of serendipitous times when the work that one half of the group is doing impacts on what the other half is doing. So we have these small molecules that we knew messed around with splicing factors in the context of senescence. So we said, oh, okay, what happens if you put them on the cells? And what happens if you put them on our human beta cells? You can throw as much glucose at those cells as you want and they don't shift. So this tells you that the splicing repertoire is really important and actually fundamental for maintenance of cell identity in this case. So what we're doing now, and this is a really big ask, but we've just got the transcriptome back and I'm very excited. We haven't written it up or done anything with it yet, so watch this space. But so I've got a very good postdoc who has managed actually to isolate those green cells from our beta cells, which is really, really hard because they're about 3 to 5% of our beta cells have changed.
Okay.
And she's fact sorted them, so she sorted them with an internal marker. So we've had to blow the cell membranes open to get the antibodies in to be able to sort them. And then I've asked her to get a transcriptome out. So it's, when we mentioned we were going to do this, a lot of people who do a lot of fact sorting kind of suck their teeth at us like a mechanic would if you bring in your broken car. But she did it. So we've just got the data back for that. So we're looking at what the precise transcriptomic changes are, and scores on the doors. I can't say too much because we haven't done too much, but let's just say that splicing is writ big.
Okay, that's good to hear that. Excellent, excellent. So coming back to sort of the future now, I noticed that you're on the schedule for next year. It's April, the Longevity Leaders World Congress, which is
Indeed.
the biggest thing in the longevity biotech space. Your talk is going to be on the role of alternate mRNA processing regulation of age-related genes. That'll be April 2020. I pose this question to everybody because I always want infinity dollars to fall out of the sky to fund your work. But take us out, if you would, as far as you were comfortable, 2025, 2030. I mean, where do you see all this going? Where do you want to be in 10 years in terms of therapeutics, other intervention modalities, and so forth? Take us on a little trip.
Okay, 10 to 15 years, I want something in clinic. That's pretty much it. So most of the efforts that we're making now, we're going after a few indications where we're developing, or we're hoping to develop some of these interventions that we've shown in cells. We want to basically take that forward in a drug development arena.
Okay.
So we're looking at identifying. We have some targets. I can't say too much about them because they are still under test, and we're got stuff happening, but we have some targets that we are going to be developing some of these RNA therapeutics too, and then we're going to be taking them into some low-hanging fruit, if you like, for indication. And hopefully that's going to happen in the next sort of 5 years or so. And what we're wanting to do is to exploit those targets, develop some novel RNA therapeutics, and get them into trials. So that's where we're going. How long it's going to take us? These things are never as clear. They're always easier to say than they are to do. And I'm sure there are going to be some bumps along the road. But I'd be hopeful that certainly in the coming decades we will have some novel small molecule or RNA therapeutic interventions for specific diseases of aging. That's where we're headed.
Excellent, excellent. One final question, getting back to you, and this is typically a wrap-up question we give regarding sort of influencers along the time that you've been doing your fascinating work. Obviously, you've stayed with this, and as mentioned, this is a very, let's call it esoteric, but it is more an esoteric area of the biotech scene. Have there been people, obviously you met a bunch of fascinating people along the way, but have there been specific people that you may want to give a shout out to here that kept you on this path, that, when things were maybe not looking great in the lab during experiments, you wanted to leave, but said, stay with this? Anyone?
Sure, yeah. I mean, like anything, we all stand on the shoulders of giants, right? Yeah. So I would say from my own personal journey, the support I had in re-establishing myself in science after a 6-year career break, I had some amazing support from here at the University of Exeter Medical School. So name check Rna. She'll hate me for doing that, but she was one of the people who was my supervisor and my mentor as a returning postdoc, and she's just been amazing. In terms of my journey on the aging research, I've been very inspired by Judy Campisi's work, as everybody has, and all of the sort of seeing what's been happening with senescent cells and the realization that senescence is a real thing. So I think learning from the work that she's been doing has been really inspirational. again, I have to give David Melzer a name check. So he's an epidemiologist and population public health person here at Exeter, who is responsible for getting me into aging. So my journey into aging came about because of one of those chance conversations in the coffee shop where he said, I've got all of this expression data from 800 people in blood. I want to look at age. I don't really know what to do with it. Want to help? And that's what got me into that. And my amazing team, I would say, you know, when I've been speaking at a lot of events, including the one in April that's coming up. There's a few more as well. And I stand there and I present their data. Right. Mm-hmm. So my job these days is to source funds to support the team and to come up with wacky ideas that sometimes work and sometimes don't. These are the guys on the ground that have done the work, and it's hats off to them. It's their work. So there, I'd particularly like to name check Eva Latour, who was the postdoc that did all of the cell reversal work, and Ben Lee, who is taking it forward now, and he's the person that's sort of moving that forward, and Nicola Jeffrey, who's done all of the cell identity work. So she's an absolute genius with cells. You know, she's a cell whisperer. I don't know how she does it. She gets things working that people just say is impossible. Also, the other members of my team, there's Jed Lai, who was an MSc student who's just had a paper out, so go read it. It's very interesting. He's been looking at astrocyte senescence and cognitive dysfunction. We've also got a clutch of new PhD students. So Laura Bramwell, she's looking at drug repurposing in the context of senescence. We have Ryan Frankham and Imad Mani, who are looking, again, they're going after some of these RNA therapeutic targets. I've probably forgotten some people. But yeah, my team, they are the people who have done the work, and you know, without them, none of this would have been possible.
Wonderful, wonderful. I appreciate that, and you know, once again, really thanking you for what you do, thanking you for championing some of these more difficult areas, swimming against the tide in certain ways, because I said earlier on, it's all about complexity, and aging and chronic disease is extremely complex, and we need to have people like you at all, you know, at all parts.
So you talk about complexity. So another example, another person in my team, Shanaz Hack, she works on yet another class of newly discovered RNAs which regulate the regulators that regulate the regulators. So she's got a paper that's coming out on aging and circular RNAs.
Circular RNA, there we go.
Circular RNA. That's going to be coming out in Geroscience any minute. So again, you know, without her and the rest of the team. So yeah, it's their work. I'm only the person that shouts about it.
Well, thank you for championing it all and leading this wonderful team. And for everybody that's going to be watching or listening on the radio stations, we've been spending time with the amazing Professor Lorna Harries, Professor of Molecular Genetics at the University of Exeter Medical School. She's also head of the RNA-mediated mechanisms disease group there. And yeah, as she said in our recent article, she wants to shout about RNA therapies. Well, I think we should all shout about Professor Lorna Harries, because clearly leading a most important endeavor. And as we say on the show, thank you for leading the human story forward in everything you do.
Oh, you're very welcome.